This research was conducted by Michelle Mendoza and Dr. Ruben Alarcon. Its focus was on the effects of a nutritional supplement on brood production in honeybee colonies. Honeybees are a major pollinator for the agricultural industry, but this is very concerning because their population is on the decline. However, the exact cause is still unknown. There could be many factors like parasite infections, malnutrition, or poor beekeeping techniques. Honeybees with a healthy nutritional diet were found to fight better against pathogens and diseases. In this experiment, a nutritional supplement with vitamins and essential oils were fed to test the reproduction of these honeybees. Half were tested with a nutritional supplement and the other half with a sugar syrup. They were both monitored by the number of cap, brood, and larvae produced. We hypothesized that the colonies fed with the nutritional supplement would have a better outcome. The start of this experiment was from September 12th through October 10th, 2020, with 24 colonies. 12 of the colonies were given the nutritional supplement, and the other 12 were given the control of sugar syrup. The colonies were from a beekeeper in Camarillo from a local ranch where they were fed a quart of either a nutritional supplement or sugar syrup once a week. In the beginning of the experiment, photos were taken of the two innermost frames of cap, brood, and larva. Every 3 weeks, additional photos were taken from the same two frames labeled on each side either the letter A or B for a total number of 3 sampling dates. The photos were taken from a camera 1 meter away from a frame holder which was attached to an empty standard hive body. We then manually counted the number of cap, brood, and larva for each frame size using image J. After we calculated the total number of brood produced for each sampling day and analyzed these data using repeated measures ANOVA. For the results, we looked at 3 factors that could determine a difference in reproduction between the nutritional supplement and sugar syrup, which were cap, brood cells, larvae, and the total brood. First, looking at the cap, brood cells, there were no significant difference between the feeding treatments nor in the time effect. In regards to the number of larvae, there was no significant difference between feeding treatments, but there was a significant difference in the time effect. In total brood, meaning cap, brood, and larva, there was no significant difference between treatments, but there was a significant difference in time effects. Even with the addition of B vitamins in the nutritional supplement, which was found to be beneficial to their immune response and protect against viruses, there is no difference between the supplement fed and the control colonies. Contrary to our beliefs, there was no difference between the supplement fed and control. This could have been due to low sample size. Further study is still needed to quantify the effects it has on brood production. If it does, beekeepers could incorporate it into their feeding regimen to foster healthier colonies. We would like to thank Wayne Scott, Shirley Williams, and Kevin Brock for helping to maintain the honeybee colonies. And thank you to Dave Mitchell, who let us use his colonies for this experiment, and Broom Ranch for allowing us access to their property.